Good morning, student. Myself Manoj Chotia from Raghunath Vidyalaya. We are studying genetics. In genetics, we studied about Mendel's life cycle. Mendel chose the pea plant for his experiment, and his experiment monohybrid cross, dihybrid cross, and polyhybrid cross. Among them, mono and dihybrid cross are our in our course. And today we are discussing the law of Mendel. Mendel's law of inheritance, and these laws are also called law of genetics. So question comes just like write down the law of genetics or the law of Mendel's law of inheritance. On the basis of two experiments, Mendel gives three rules. First, law of dominance, second, law of segregation, and third, law of independent assortment. So there is 1 mark question preparing here. First of all, on which experiment law of dominance is depend, or law of segregation is depend, or law of independent assortment is depend. So the first two law of dominance and law of segregation are depend upon the monohybrid cross, and the law of independent assortment is depend upon the dihybrid cross. First of all, we are looking law of dominance. Whenever we cross two characters, listen carefully, we are taking example. This is not rule. Whenever we cross between tall plant and dwarf plant, then in F1 generation only tall plants comes. Here this is tall and this is dwarf, and this is F1 generation. In F1 generation only a single character, means here there are two traits. The character is height of plant, and height is two traits, tall and dwarf. Whenever we cross between tall and dwarf, then in F1 generation only a single trait comes, and that is tall, we know about. But Mendel did the experiment on it. So the law is state that the character which is present in F1 generation, the character which is present in F1 generation is called dominant, and the character which is not present in F1 generation is called recessive. So this is the law of dominance. The character which present in F1 generation is called dominant, and it is dominant on this recessive character, and this is called law of dominance. So it is the example. हम दूसरा एग्जाम्पल भी ले सकते हैं, जैसे राउंडेड सीड और रिंकल्ड सीड में क्रॉस करवाएंगे तो वो नियम नहीं हुआ, यानी लंबा यहाँ F1 जनरेशन में जो टॉल प्लांट आ रहे हैं, दैट इज नॉट लॉ. The law is that in F1 generation which character is present, that is dominant character, and the character which is not present in F1 generation is called recessive, and this is called law of dominance. अब यहाँ पर ले सकते हैं क्या, एग्जाम्पल. ये अपने पास हो जाएगा एग्जाम्पल. और अगर आपके लॉ ऑफ डोमिनेंस आता है, 2 मार्क का क्वेश्चन आता है, तो आपको F2 जनरेशन तक इसमें रेशियो निकालना है. जैसे अब यहाँ आगे क्या होगा कि F1 जनरेशन का क्रॉस किससे करवाते हैं, F1 से. F1 जनरेशन का क्रॉस F1 से करवाएंगे तो क्या प्राप्त होगा अपने को, ये अपन पहले पढ़ चुके. तो यहां अपने को क्या प्राप्त होगा, ये रेशियो प्राप्त होगा, यानी फिनोटाइप इज ऑब्टेन्ड 3:1 एंड जीनोटाइप इज ऑब्टेन्ड 1:2:1, and this one is pure and this is hybrid. These are the hybrid tall, and this is pure tall, and this is dwarf. Pure tall, hybrid tall and dwarf. So यहां तक अपने को निकालना है. अब ला क्या हुआ कि whenever we cross between two characters or the trait of two characters, then the character which present or show in F1 generation is called dominant, and the character which is not present in F1 generation is called recessive, and this is the law of dominance. Whenever we look at the importance of organism or in human genes of related to law of dominance, then we see that there is so many heredity disease are recessive in presence of dominant gene, cannot be expressed themselves, hence the human remains normal. Like diabetes, color blindness are the diseases which present through heredity, but all the genes are present in human being, but they are not shown. So with the help of law of dominance, and this law is depend upon the monohybrid cross. Now second, law of segregation, and this law is also called law of purity of gametes. Law of purity of gametes. This law is also called law of purity of gametes. And यहाँ एक नंबर का क्वेश्चन फिर बनता है कि why law of segregation called law of purity of gametes. So you have to remember first of all what is the law of segregation. Before crossing means the inheritance law which is given by Sutton and Boveri. What is the law? The characters, the gene or the chromosomes which are present in the form of pair, means all the chromosomes or the genes present in any organism in pairs. And what is the law? Law is that before crossing these pairs segregate from each other. If we show the presentation of law of segregation, then it is the presentation. क्रॉस करने से पहले क्या हो गए? ये सेपरेट हो गए, ठीक है. दिस इज द लॉ ऑफ सेग्रीगेशन. और अपन ने यहां देखा, देखिए F1 जनरेशन का क्रॉस जब F1 से करवाते हैं तो अपने पास ये क्या था? TT. और ये भी क्या है? सेग्रीगेट हो जाते हैं. अब ये क्या है? गेमेट. और गेमेट क्या है? मैंने आपको बताया हुआ है, द स्ट्रक्चर इन विच क्रोमोसोम्स नंबर ऑफ क्रोमोसोम्स रिमेन्स हाफ इज कॉल्ड गेमेट. तो इस स्टेज में इसको अपन क्या, यहां जैसे अपन ने बात की थी कि ये दोनों P कैपिटल है, तब ये क्या हुआ? प्योर टोल. और ये दो कैसे हैं अपने पास? ये है हाइब्रिड टोल. ये कैसे हैं? हाइब्रिड टोल. और अगर अब अगर मैं आपसे ये पूछूं कि ये सिंगल टी किसको डिनोट करता है? ये सिंगल टी किसको डिनोट करता है? सीधा सीधा आंसर होगा टोल को. और ये किसको डिनोट करता है? तो ये करता है किसको? ड्वार्फ को. तो इस समय हम at the time of segregation we can't say the pure or the hybrid word. We can't use pure and hybrid word. So in the stage of gamete they are pure. So this is the law of segregation. And what is the significance of law of segregation? So first of all, gene hypothesis proved by this law. Gene hypothesis proved by this law. And gene is the unit of inheritance which transmit characters from one generation to another generation. Third, each character is controlled by gene in organism. Each character is controlled by gene in organism. And fourth, each gene has two alleles, means genes present in the pair. Here they are present in pair, here they are also present in pair. So each gene has two alleles which control single pair of contrasting character, which control single pair of contrasting. And next, the allele of gene remains together but do not mix with each other. यहाँ ये mix नहीं होते, so they do not mix with each other. And next and last significance, alleles become separated from each other during formation of gamete. Alleles become separated from each other during gamete formation and transfer in separate gamete. So this is the significance of law of segregation. Now third, the law of independent assortment. in starting of this video, I tell you that the law of independent assortment is depend upon the dihybrid cross. And what is the law of segregation, independent assortment? You can see properly by this example. In dihybrid cross, we make or we take two characters. We take two characters and cross them with their recessive characters. For example, listen carefully. I am telling you once again. We are taking example. What is the example? Tall and rounded seed. ये प्लांट कैसा है? टोल है जिसमें राउंडेड सीड आते हैं. इसका क्रॉस अपन करवाते हैं किससे? ड्वार्फ और रिंकल्ड सीड से. ठीक है. तो यहां फ्रॉम द लॉ ऑफ डोमिनेंस क्या हुआ अपने पास? ये सारे के सारे क्या है अपने पास? ये सारे के सारे क्या है? टॉल एंड राउंडेड सीड वाले प्लांट आए. ठीक है. ये F1 जनरेशन. अब देखिए लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट क्या है? जनरेशन. ये जो करेक्टर है कौन सा? ड्वार्फ एंड रिंकल्ड सीड. ये करेक्टर यहां नहीं आया. ठीक है. ये करेक्टर यहां नहीं आया. तो क्या हुआ? द करेक्टर विच इज नोट कम्स इन F1 जनरेशन which is not destroyed and they have their own strand which present in F2 generation. This is the law of independent assortment. Means ये करेक्टर यहां प्रेजेंट नहीं हुआ. अब अपने को पता है कि F1 जनरेशन में रेशियो क्या है. तो कोई भी रेशियो अपन क्या कर देंगे? 1:0. अब देखिए F2 जनरेशन में आप लोगों को याद होगा अपन ने डाई हाइब्रिड क्रॉस पढ़ा था. क्या रेशियो आता है? 9:3, 3:1. ये रेशियो है. अब इसमें से 9 कैसे है? ये 9 तो कैसे हैं? टॉल एंड राउंडेड है. ठीक है. 3 कैसे हैं? 3 है टॉल एंड रिंकल्ड. और फिर 3 कैसे हैं? फिर 3 है ड्वार्फ एंड राउंडेड. और 1 कैसा है? 1 है ड्वार्फ. ये आप लोग पीछे का वीडियो देख लेना, उसमें आपको क्लियर हो जाएगा. ये जो 9:3, 3:1 रेशियो आते हैं. तो इनमें देखिए अब यहां इसको तो छोड़ दो. यहां क्या आया? रिंकल्ड. कितने? 3. यहां ड्वार्फ कितने? 3. यहां एक ड्वार्फ और एक रिंकल्ड. मींस 8 अपने पास क्या आ गए? रिसेसिव. 8 क्या आ गए अपने पास? रिसेसिव कैरेक्टर्स विच इज नॉट प्रेजेंट इन F1 जनरेशन. सो दिस इज लॉ ऑफ इंडिपेंडेंट assortment. It is clear from the above experiment that beside the parental combination of tall and round seed and wrinkled seed present, 2 new combination of dwarf and wrinkled seed are present in F2 generation. This is called Mendel's law of independent assortment. क्या हुआ? सीधे सीधे शब्दों में आपको बता देता हूं कि डाई हाइब्रिड क्रॉस में जो करेक्टर F1 जनरेशन में प्रेजेंट नहीं हुए थे, यहां ये करेक्टर अपन ने लिए थे टॉल एंड राउंडेड. ये एग्जांपल है, फिर बोल रहा हूं. और ड्वार्फ एंड रिंकल सीड, तो ये यहां प्रेजेंट नहीं हुए थे. तो ये प्रेजेंट नहीं हुए, इसका मतलब ये नहीं है कि ये डिस्ट्रॉय हो गए. इन्होंने अपना क्या रखा? इंडिपेंडेंट असॉर्टमेंट रखा. और ये इंडिपेंडेंट असॉर्टमेंट F2 जनरेशन में प्रेजेंट हो गया. दिस इज द लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. सो सिग्निफिकेंस क्या है? विद द हेल्प ऑफ एक्सपेरिमेंट Mendel declared all the law of principle given earlier scientists because most of them were only fictional and they have not only scientific base. In the beginning, Mendel's work had no recognition, but by this law, the new experiment and discoveries in genetics were achieved. So today we studied about law of inheritance or law of Mendel, or we can say law of genetics. Which are the law? First, law of dominance, second, law of segregation, and third, law of independent assortment. Thank you for today's class.